Hello everyone. Welcome back to Microbial Engineering and Algal Biotechnology Laboratory, Department of Biosciences, JIS University, Kolkata. Today we are going to discuss on impacts of current vaccines on COVID-19 variants. The entire project has been conceptualized by Dr. Deepankar Ghosh, Assistant Professor, Department of Biosciences, JIS University, Kolkata. First of all, I'd like to put some notes for our viewers. The MEAB channel is the platform to nurture students' knowledge and motivate them to carry out proper literature survey as the innovation gap filler for their future progression under the great umbrella of JIS University and JIS Group, where PI of MEAB Laboratory has been engaged at present. Moreover, MEAB channel would like to spread awareness for the community services. So this platform is not to publish research work as such. We believe that there are plenty of quality journals and books available where we do publish our scientific work to reflect our smart work. The MEAB group believes on quality, not on quantity. We do not compete with other research communities. Instead, we intend to put hands together for the progression of great science and technology. Thank you all. So first of all, I would like to congratulate and also welcome Shubhangi, Paramjit and Sampriti for joining in my lab as a BSc Summer Interns Research Project student in the year of 2021. And I would say they are the cream of the future in my laboratory. Thank you, all of you. So let's get started with the topic of today's deliberation. So I will get started with the progression of this research topic. And finally, Shubhangi, Paramjit and Sampriti will be continue rest of the presentation. So let's get started the glimpse of the impact of current vaccines on the different variants of SARS-CoV-2. Upon the emergence of the newest pandemic, COVID-19 vaccine, and the suitable drugs or chemotherapeutics have been much awaited as COVID-19 is much more than an health crisis. It leads to a political, social, economic crisis in the entire globe. The development of safe and effective vaccine might effectively help bring the situation under control. For an infection that is the viral or related to the respiration organs cannot be fully prevented by vaccination, but it strengthens the immune response and immune system to an extent where life risk of an individual is lesser. The induction of the alterations during replications are known as mutations, and a virus with one or more mutations is referred to as a variant. So the more changes in the viral genome undergoes as it spreads more its opportunity to replicate. The changes in the virus's genetic material do affect its property, the phenotypic traits, such as the rate of transmission, severity of the disease progression, and also that will induce the chances of infection in a healthy individual. So in this presentation and current context, we are going to discuss and illustrate about the vaccine currently available in the local and the global scenario and the market to prevent or eradicate the further spread of COVID-19 at current state. So their mode of action in the molecular basis and their probable effects on the disease will also be discussed. According to the World Health Organization, WHO, at least 4 different types of coronaviral variants are spreading through the globe since the first case in November 2019. The strains are being classified like VOC, the variant of concern, VOI, the variant of interest, VUM, the variant under monitoring. So far, there have been over 7 identified variant cases have been implicated. They do infect the people worldwide. So rest of the part of the deliberation will be continued by Shubhangi, Paranjit and Sampriti. So the dice is over to them. Thank you.
So far, multiple variants of coronavirus have been identified worldwide. The Alpha strain was identified at Kent, UK in December 2020. The Beta strain was identified in South Africa in October 2020. The Gamma strain was discovered in Brazil, December 2020. The Delta strain was identified in Maharashtra, India in October 2020. The Epsilon strain was identified in California, September 2020. The Eta strain was identified in New York in November 2020. The Kappa strain was first detected in India in the recent times. while the Lambda strain was first detected in Peru in December 2020. Given the severity of the disease and the multiple variants that had been detected over the time, multiple vaccines have been developed for the drive against SARS-CoV-2. Some of them are as follows. The Pfizer vaccine was developed by BioNTech, the German company. It partnered with Pfizer for the development of the vaccine, and the vaccine came to be known as BNT162b2. The generic name was tocilizumab and the brand name is Comirnaty. The Moderna vaccine was developed by Moderna TX Incorporated, an mRNA vaccine which was known as mRNA-1273. The Oxford-AstraZeneca vaccine, as the name suggests, was developed by the University of Oxford and its spin-out company, Vaccitech. The Sputnik V or Sputnik 5, developed by Gamaleya Research Institute in Russia. The Janssen vaccine was developed by Janssen Vaccines in Leiden, Netherlands, and its Belgian parent company, Janssen Pharmaceuticals, a subsidiary of the American company Johnson & Johnson. While the Covaxin was developed by Bharat Biotech in collaboration with Indian Council of Medical Research ICMR, National Institute of Virology NIV. Now we'll delve deeper into the understanding of how these vaccines work, starting with the Pfizer vaccine. This vaccine requires 2 shots given at an interval of 21 days. It is developed based on the genetic information of the virus for the spike protein. It uses an mRNA wrapped in oil bubbles made up of nanoparticles in order to prevent the mRNA from degradation by the natural enzymes of our body. The vaccine particles fuse to the cells of our body when injected and release the mRNA. The cells read the sequence of the mRNA to synthesize the spike protein, and the cells eventually destroy the mRNA. The spike protein either get presented on the surface of the cell after fragmentation, or they migrate to the surface and stick out their tips. These protruding tips or the fragments are recognized by the immune system. Antigen-presenting cells then take up the spike protein debris when the vaccinated cells die and present them on the surface. The helper T cells then detect these fragments, which then recruit the other immune cells to fight the infection. Other cells of the immune system, like the B cell, also encounter the spike proteins on the surface of the vaccinated cells, or free-floating protein fragments. The B cells on activation start to proliferate and produce antibodies targeting the spike protein. The antibodies bind to the spike protein and prevent it from binding to the cells in the body and mark the virus particles in the body to be destroyed and prevent infection. The cytotoxic T cells recruited by the antigen-presenting cells also recognize and destroy the cells that display the spike protein on the surface after being infected by the coronavirus. The recent findings have suggested that the RNA vaccines which have been currently made available in the market are potent against the immune-evading variants. Both Pfizer and BioNTech are currently developing upgraded RNA-based vaccines to target the beta variants of the virus. We will now discuss the mode of action of the Pfizer vaccine. As mentioned previously, it uses an mRNA wrapped in oil bubble made up of lipid nanoparticles so that it does not degrade when in the action of the natural enzymes present in our body. After this, it's injected into the body where the virus particles fuse to the cells and release the mRNA. The molecules of the fused cells follow the sequence of mRNA and build up the spike protein, and post this, the mRNA dissolves, leaving behind no trace. Now, if the vaccinated cells were to die, the debris would have spike proteins and protein fragments which can be taken up by antigen-presenting cells. When helper T cells detect these fragments on the cell surface, it alarms the other immune cells to help the body fight the infection. The B cells may build a protection against COVID alike by the spikes on the surface of vaccinated cells or free-floating fragments by getting attached. If these B cells get activated by the helper T cells, they start to proliferate and pour out antibodies targeting the spike protein. The antibodies can then also latch onto the coronavirus spikes, leading the virus to destruction, and further leads to prevention of infection by blocking the spikes from attaching to the cell. The antigen-presenting cells can also activate killer T cells to destroy affected cells that show similar kind of spike protein fragments on their surface. We will now discuss about the Moderna vaccine.
Moderna Vaccine. It requires 2 shots which are 20 days apart given as shot in the muscle of the upper. The researchers have also observed that the Pfizer and Moderna vaccine would provide a sturdy protection for at least a period of 12 weeks after the administration of the second dose of the vaccine and could also provide a low-level protection for at least a year. It was reported by Moderna on 29 June 2021 that the vaccine produced neutralizing titers in response to the tested variants, which include the beta variant first identified in South Africa, 3 lineages variant of B.1.617 first identified in India, including the Kappa and the Delta variants, and the Eta variant first identified in Nigeria, and the A.23.1 and A.VOI.V2 variant first identified in Uganda and Angola, respectively. In the Moderna vaccine, the nucleoside-modified mRNA is constructed in lipid particles, which allows their delivery into cells of the host, which allows the expression of the spike antigens of the coronavirus. Thus, the vaccine elicits an immune response to the spike antigens, which contributes to the protection against COVID-19. The recent studies on Pfizer and Moderna vaccines has revealed that the vaccine may offer immunity against the virus for a period longer than it was initially expected. Scientists under the leadership of Ali H. Elibedi, PhD of the Department of Pathology and Immunology at Washington University School of Medicine in St. Louis, have investigated and found that the immune cell responses in the germinal center of the lymph nodes after a single dose of Pfizer vaccine have shown to remain active longer than expected. This intimates the possibility of the mRNA vaccines offering an extended protection against the coronavirus. Oxford-AstraZeneca vaccine. It requires 2 shots administered 4 weeks apart. The Oxford-AstraZeneca vaccine, condemned as AZD1222, while Covishield and Vaxzevria are its brand names. It's administered intramuscularly via an injection. The vaccine uses modified chimpanzee adenovirus CHAdOX1 as a vector. The efficacy of the vaccine was found to be 76% at preventing symptomatic COVID-19 following the first dose and 81.3% after the second dose. The vaccine has been found to be 66% effective against the Alpha variant, while 60% effective against the Delta variant. Its side effects include injection site pain, headache, and nausea, which resolve within a few days. It has been approved by European Medicine Agency (EMA) and Australian Therapeutic Goods Administration and the World Health Organization for emergency use listing. The vaccine has been authorized for use in more than 70 countries, AstraZeneca has stated. So here's the mode of action of Oxford-AstraZeneca vaccine. That is, how does Oxford-AstraZeneca vaccine works? The vaccine uses a modified version of a chimpanzee adenovirus CHAdOX1, which can enter a cell but it cannot replicate inside the cell. Upon injection, the adenoviruses bump into the cells and attach onto protein surface. The cell engulfs the virus in a bubble and pulls it within. In the cell, the adenovirus escapes from the bubble and moves to the nucleus towards the cell's DNA. The adenovirus DNA is pushed into the nucleus where it is copied into messenger RNA or mRNA. The mRNA then leaves the nucleus and then the cell and starts assembling spike proteins upon reading its sequence. Some of the spike proteins migrate to the cell surface. Some of the proteins of cells are also fragmented. The producing spikes and protein fragments are recognized by the immune system. The immunity is provoked by the immune system to reacting to the spike proteins. Upon death of a vaccinated cell, the debris contain spike proteins and protein fragments that can be taken up by the antigen-presenting cells. T cells detect the fragments and help develop immune cells to fight against the infection. B cells sometimes lock into the spike proteins, and when activated, they produce antibodies that target these spike proteins. The antibodies prevent infection by blocking the spikes from the attaching to other healthy cells. T cells seek out and destroy any cell that display the spike protein fragments on their surfaces. Sputnik V requires 2 shots. It has 92% efficacy against the original strain and 90% efficacy against the Delta strain. The vaccine uses a weakened virus to deliver small parts of a pathogen and stimulates an immune response. It is a vector vaccine-based adenovirus in which the coronavirus gene is integrated, a 2-component vaccine in which adenovirus serotypes 5 and 26 are used. It has been authorized for distribution in 68 different countries. Some are as follows: Belarus, Argentina, Algeria, Bolivia, Serbia, Palestine Territories, Mexico, DGCI, Bangladesh, Turkey, Albania have taken it for emergency use. Czech Republic, Berlin, Filipinos have given it full authorization, and European Medicine Agency has kept it under review. The most common side effects were injection site pain, injection site swelling, tiredness, headache, muscle pain, joint pain, fever, diarrhea or vomiting that resolves within a few days. So here's how the Sputnik V works. The Sputnik V vaccine is comprised of adenoviruses AD26 and AD5, where the coronavirus spike protein genes are added, capable of invasion but not the replication. So upon administration, the adenoviruses fuse to the cells and attach onto the protein surface. The cell then engulfs the virus. Once inside, the adenovirus travels to cell's DNA. The adenovirus pushes its DNA into the nucleus. The adenovirus is unable to replicate, but the gene for the coronavirus spike protein can be read by the cell and copied into mRNA. When a vaccinated cell dies, the debris would have spike proteins and protein fragments in it, which can be taken up by an antigen-presenting cell. When helper T cells detects these fragments on the cell surface, they can alarm, helping other immune cells to fight against the infection. The B cells may build protection against coronavirus-alike spikes on the surface of a vaccinated cell or free-floating fragments by getting attached to it. If this gets activated by helper T cells, they start to proliferate and pour out antibodies targeting the spike protein. The antibodies can latch onto the coronavirus spikes, leading the virus to destruction and prevent the infection by blocking the spike from attaching to the other cell surfaces. So the antigen-presenting cells then activate the killer T cell to destroy the affected cell that shows same kind of spike protein fragments on their surface.
Next, we come to India's first indigenous COVID-19 vaccine, that is Covaxin. Covaxin requires 2 shots with a time lapse of 28 days in between. It has been found effective against both Alpha and Delta strain according to the US National Institute of Health. The vaccine has been developed using whole virion inactivated virocell derived from platform technology, included along with the immunopotent immune potentiators, also known as vaccine adjuvants, for increasing and boosting its immunogenicity. The vaccine has the authorization from India's Drug Regulatory Authority, the Central Drugs Standard Control Organization, and was authorized for emergency use on January 2021. Covaxin is an inactivated COVID vaccine made out of a sample of the coronavirus isolated by India's National Institute of Virology. It helps the immune system to make antibodies against the coronavirus which attach to viral proteins or the spike protein. The possible side effects of the vaccine reported are fever, headaches, irritability, pain, swelling, or both at the site of injection and are expected to be shown for a few days after vaccination. The detailed mode of action is discussed in the next slide. The isolated coronavirus sample is first treated with beta-proteolactin that binds to viral genes, disabling the replication property of the virus, keeping the spike proteins intact. A little amount of aluminium-based adjuvant is mixed with inactivated virus. When Covaxin is shot, the antigen-presenting cells in recipient's body swallow up the inactivated cells, tear the coronavirus apart, and take up the spike proteins on the surface. When helper T cells detect these fragments on cell surface, the helper T cells can alarm, helping other immune cells to fight the infection. T cells may encounter the inactivated coronavirus, and the B cell surface proteins, if latch onto the coronavirus, pull the virus inside and show coronavirus fragments on its surface. If these B cells get activated by helper T cells, they will start to proliferate and pour out antibodies targeting the spike protein. Once vaccinated with Covaxin, the immune system responds against live coronaviruses. B cells produce antibodies that stick to the invaders and target the spike protein and can prevent the virus from entering cells. Bharat Biotech's phase 3 clinical trial demonstrates that Covaxin protects people against COVID-19. Researchers will have to observe it for a few months to see how long that protection lasts. It's possible that the level of antibodies will drop, but the immune system also contains special cells called memory B cells that might retain information about the coronavirus for years or even decades. Next, we have the Johnson & Johnson vaccine against COVID-19, or the Janssen vaccine. It is a viral vector vaccine based upon the human adenovirus, modified to contain the gene for making the spike protein of SARS-CoV-2. The body's immune system responds to this spike protein and produces antibodies. The vaccine requires only one shot and need not to be stored in frozen conditions. Clinical trials for this vaccine started in June 2020, and the phase 3 trials involved around 43,000 people. Janssen announced that the vaccine was 66% effective in a one-dose regimen in preventing symptomatic COVID-19 and showed 85% efficacy in preventing severe COVID-19 and about 100% efficacy in preventing hospitalization or death caused by COVID-19. The vaccine has been approved for an emergency use by the US Food and Drug Administration and a conditional marketing authorization by the European Medicines Agency and the UK Medicines and Healthcare Products Regulatory Agency. The vaccine is used to provide protection against the infection by SARS-CoV-2 virus in order to prevent COVID-19 till aged 18 years and older. It is given intramuscularly via injection into the deltoid muscle. One dose is sufficient, and evidence that a booster dose is needed has been lacking. The vaccine is based on the principle of replication incompetent recombinant adenovirus type 26 vector expressing the SARS-CoV-2 spike or the S protein in a stabilized conformation. The vaccine contains the following inactive ingredients, which include sodium chloride, citric acid monohydrate, polysorbate 82-hydroxypropyl beta cyclodextrin or HBCD, trisodium citrate dihydrate, ethanol, hydroxypropyl beta-dex, sodium hydroxide and hydrochloric acid. Next, we have the mode of action of the Janssen vaccine. The vaccine is comprised of adenoviruses, where the coronavirus spike protein genes are added and are capable of invasion but not replication. Upon administration, the adenoviruses fuse to cells and attach onto the protein surface. The cell then engulfs the virus, and once inside, the adenovirus travels to cell's DNA. The adenovirus pushes its DNA into the nucleus, and it is unable to replicate, but the gene for coronavirus spike protein can be read by the cell and copied into mRNA. When a vaccinated cell dies, the debris would have spike proteins and protein fragments, which can be taken up by an antigen-presenting cell. When helper T cells detect these fragments on cell surface, they can alarm helping other immune cells to fight the infection. B cells may build protection against coronavirus like spikes on the surface of vaccinated cells or free-floating fragments by getting attached. If these get activated by helper T cells, they start to proliferate and pour out antibodies targeting the spike protein. The antibodies can latch onto coronavirus spikes, leading the virus to destruction and prevent infection by blocking the spikes from attaching to other cells. The antigen-presenting cells can also activate killer T cells to destroy affected cells that show same kind of spike protein fragments on their surface. So how efficacious are the vaccines against the variants? Variants remain to be a major cause of concern among scientists because these mutations enable the virus to survive and evade the antibodies in someone's bloodstream, thereby infecting more people and putting more lives at probable risk. It has been noted that there are 3 mutations in the epsilon coronavirus spike protein that reduce the effectiveness of the antibodies generated either by vaccines or past COVID infections. After testing the resilience of blood plasma from vaccinated people and people who had already had COVID, researchers found that the ability to neutralize the epsilon variant has reduced to 2 to 3.5 times The Delta Plus variant has been formed as a result of the Delta variant acquiring a mutation called K417N in the spike protein, according to the New England Journal of Medicine. The spike proteins on the SARS-CoV-2 Lambda variant have shown a twofold increase in infectivity, which has been predicted due to a mutation on the virus called the L452Q mutation. When it comes to efficacy of vaccines, it has been suggested that optimal immunity is established when 2 doses of the AstraZeneca vaccine are given 3 months apart, which is much longer than the recommended 3 weeks for the Pfizer vaccine. The finding corresponded with the data from the Francis Crick Institute published in The Lancet. Johnson & Johnson has stated that its single-shot vaccine neutralizes the Delta variant and provides durable protection against the infection more broadly. The Sinovac Biotech Limited's CoronaVac vaccine has been administered alongside with the Pfizer and the BioNTech shot in Chile, and substantial difference has been found in protection against symptomatic COVID at a time when Alpha and Gamma were variants most frequently detected. It has been mentioned that both inoculations provide a high level of protection against severe diseases and death, according to a study published in the New England Journal of Medicine on July 7.
Well, we come to a conclusion. The COVID-19 pandemic, which started in the beginning of 2020, has been a bigger cause of concern in the last 16 months. Large-scale studies, vaccine drives have been underway even since the virus broke out, causing havoc and mass morbidity and mortality in the entire globe. The world is yet to learn, comprehend, and understand the severity of the pandemic, with newer variants and variants of the virus emerging, re-emerging day by day. The studies have conducted so far suggest that the newer variants of this virus are far more hazardous and have a higher efficacy that can infect the healthy individuals. The studies on the efficacy of the vaccine are still underway. The initial results demonstrate that the vaccine should be effective against the newer strains and won't be affected by the enhanced infectivity of the virus. But there are initial observations. If the approach of the people towards safety precautions against the virus continues to be lenient, there are possibility of more numbers of strains which could pose challenges to the vaccination drive underway across the globe. So it is our moral responsibility to not only get vaccinated, also make sure like we are maintaining the proper hygienic conditions, proper sanitation each and every day. And we will also educate people, those who are around us, to maintain the proper safety precautions given by our government, given by the different health organizations for our safety, and also allow to reduce the possibility of getting infected by the natural spreading of different SARS-CoV-2 variants. So these are the references we have been collecting since last 6 months, and we have been summarizing lots of information by extracting the summary of those references. We would like to thank all of these research contributors, different doctors, nurses, and all the people, those who are serving our nation and also all over the globe, to carry out the community services to maintain our health, and also allows us to give assistance and the health all over the time. Thank you all for their great contribution. Hats off to them. Finally, we'd like to thank all our viewers, all our subscribers for their immense support and help since beginning. I hope you all have enjoyed this particular session. Thank you very much. Stay healthy, stay safe. Bye-bye.